So in our last video, we talked about proteins, what they do. Now we're going to talk about how to make proteins. So we're going to talk about protein synthesis. And proteins are synthesized by putting amino acids together, as you know from chapter 16. So how do the amino acids put together the protein? So how do we get that long peptide chain? And it comes from DNA and RNA. So DNA and RNA are composed of nucleic acids. And nucleic acids have certain parts. They have a phosphate group, a sugar, and a base. And the base is always going to be containing nitrogen. So you can see the parts here to your nucleic acid. The bases are either derivatives of pyrimidine, so a resonant structure, so an aromatic structure that looks like this. This structure has these double bonds, and then remember you have lone pairs on these nitrogens, and that causes the electrons to be able to hop around. And so this is a resonant structure. And then we have purine. And so the difference in pyrimidine and purine is that they have the same base component here, but the purine has an additional ring attached to it. So the purine bases, the double ring purine bases are adenine and guanine. And then the pyrimidine single rings are cytosine and thymine. And then RNA is the same, except thymine is replaced by uracil. And you may already know this from a biology or genetics class that you've taken. So DNA has the bases A, G, C, and T, and RNA is A, G, C, and U. So T is replaced for U in RNA. And so these either are purine bases or pyrimidine bases. The next part of the nucleic acid is a sugar, and it is a pentose sugar, which is a 5-carbon sugar. RNA, the sugar is ribose, and DNA, the sugar is deoxyribose. And the difference between the two is that there is no oxygen on carbon 2, hence the deoxyribose. And so your carbons are numbered with primes to distinguish them from carbons or atoms in the bases. So your sugars all have this like prime number system. Oh, sorry, I thought it was going to zoom in on this. So your sugars will all be numbered just like we number from chapter 13 to the right of the oxygen in the ring, carbon number 1. And so carbon 1 goes around to carbon 5. So these are your pentose sugars. And on carbon 2 is where you get your difference from ribose and deoxyribose, because ribose has an oxygen on that carbon 2, and deoxyribose does not have an oxygen on that carbon 2. So deoxy meaning no oxygen. So a nucleoside is a sugar and a base. Okay, it is the we have a glycosidic bond to carbon 1 on the sugar with the base. So we have this glycosidic bond here. So this is kind of like a culmination of our chapters. We've been talking about sugars and bases and amino acids and those units. We're all going to put it together in this chapter. And so adenine and ribose form a nucleoside called adenosine. And so here you can see this, it's a beta. Okay, so just like a sugar has a OH on top of carbon 1, we call it beta. And this is a beta connected to an N. So it's not connected to carbon 1 and carbon 4 like you would get in a saccharide. It's connected to the nitrogen. And so this is a beta N glycosidic bond. So just like how you would need to recognize an alpha or a beta 1,4, 1,6 or whatever glycosidic bond in a sugar, you would need to recognize a glycosidic bond between a sugar and a base in a nucleoside. Now nucleotides are the whole thing. We put the phosphate group in as well. And so when you have just a nucleoside, you've got just the two parts to it, so the sugar and the base. Then when we take that sugar and we also add a phosphate group to carbon 5 of that sugar, we get a phosphate ester. And so here are your nucleotides bonding. So you can see that here your nucleotide consists of the phosphate group with this kind of, it looks like an ester, right? Except for instead of a carbonyl carbon, it's a phosphate. And so this is a phosphate ester bond. And it's to carbon number 5 of the sugar. And so you can see these different bases here with different with your ribose, and then here's your deoxyribose sugar. You can see you do not have an O on that carbon 5, so they're kind of highlighting that for you. The rest of these are ribose sugars. And so your DNA and RNA comparing, right? Your nucleosides are a base in deoxyribose or a base in ribose, and then you're adding the phosphate for your nucleotide. And then a whole nucleic acid is a chain of these ribose nucleotides. And so we have pieces, and then we put them together for what we call a nucleic acid. Naming nucleosides and nucleotides is not something that I will have you do, but it is helpful just to be able to kind of get through the material to kind of have this table available if you are looking at them. I do need you to know the bases, because what we do is we only focus on the bases to look at how we code for certain amino acids and therefore proteins. So I do need you to know these bases A, G, C, T, and U. You do not need to know the nucleoside or nucleotide names for those, but just the bases. Yes, you do need to know the names of those. So do a quick check and tell me whether or not these bases are purine or pyrimidines. Here are your answers. Remember, your purine is your fused 2 rings, and your pyrimidine is your 1 ring. And then you've got other stuff hanging off of it based on what kind of base it is, right? So pause and do a quick check to see if this is if these bases are in DNA, RNA, or both. And so guanine and adenine are in DNA and RNA both. Thymine is in DNA only, and thymine is actually replaced by uracil in RNA. And so just like proteins, we have this primary structure, secondary structure stuff. You have a primary structure of nucleic acids, which the primary structures are always just putting together the chain. And so the primary structure of nucleic acid is just the chain, right? So linking together all of these nucleotides creates a chain. And this sequence of bases is what codes for things. And so the nucleic acid is a polymer, and then we're looking at how they're connected. They're actually connected at certain carbons, so let's zoom in here and see this connection. And so they're connected on carbon 5, and then the phosphate is connected to carbon 3. So we're going to talk about a 3', 5' phosphodiester bond. So you've got a bond between carbon 3' and 5' on the next sugar down. You have an end that is the 5' end. So that means that just kind of like how we had an N terminus and a C terminus for our amino acid sequence, our peptides, we have a 5' end on one end of the chain here, and we have a 3' end on the other chain. And so you look to carbon number 3 and carbon number 5 and see which one is open, not attached to anything, and that's your end. So this one is not attached, so that's our 3' end. And we have our 5' end on this side. And so we will be looking at order of things based on 5' and 3'. So their primary structure, again, is just the sequence of the bases. And so we only really focus on the sequence of those bases when we're talking about DNA and RNA, because the sugar and the phosphate are the same for every bit of DNA. It's all deoxyribose. Every bit of RNA is all ribose. And so what's different about them is the bases. So we just name them by the bases themselves. And so we call this link between them a phosphodiester bond. It is a phosphate kind of acting as an ester, but in 2 places. And so you have your double bond here, but then you have an O to a carbon and an O to a carbon. So this double oxygen is going to mean diester. So phosphodiester linkage is how we link 2 nucleic acids together. We link them together on the phosphate end of that. So the base should be sticking out at the end, not really linked to anything. The bases are actually going to interact with each other in our next structure. So you do not need to be able to draw the condensed structures, but you do need to be able to recognize that this is a primary structure, this is a phosphodiester linkage, and you have two nucleotides connected here. Again, so your nucleic acid sequence, you're only going to focus on the bases. And so we start with the free 5 end, the 5' end, and you'll say, starting at 5', adenine, cytosine, guanine, uracil, so A, C, G, U. Quick check, is this a DNA or RNA? This would be an RNA because we have uracil here. We also have the oxygen on carbon 3. Carbon 2. So carbon 2, you can see that oxygen. In deoxyribose, carbon 2 has no oxygen. So what goes in the blank is a phosphodiester bond that we're bonding here. So if we're looking at the nucleic acid listed here, which base is the free 5' phosphate end? It will be the first one because we start with the 5' as our first one, not 3'. 3' is at the end. 5' is the starting place when you're reading it. So the cytosine is the starting place. Alright, so your secondary structure here, just like the secondary structure of peptides, is going to be a helix is an option. But for DNA, it makes a double helix. So you have two strands that come together to make this double helix structure that you're familiar with in DNA. So your backbones are your sugars and your phosphate bonds. And then you have your your bases that are actually interacting with each other. And we find that the number of bases is equal in specific parts. So the number of A's, adenines, are equal to the number of thymines, or uracil if you're talking about RNA. Guanine is equal to cytosine. Therefore, we're looking at bases that are actually paired up together. So every A is paired with a T. Every G is paired with a C. So you need to know A and T go together, and G and C go together. Watson and Crick are credited with the idea of the double helix. I just want to plug out there that they had some help. There was a woman named Rosalind Franklin who actually came up with the X-ray diffraction pattern of the double helix, and someone took that from her lab and brought it to Watson and Crick, and that helped them propose this double helix, but she never got credit for it until like a long time after that. And so just know that yes, they are the ones that actually put it all together to propose the double helix, but there was another scientist, a woman named Rosalind Franklin, who her work was instrumental in helping them kind of prove that that was what it was. But they did not, of course, in that time give her any credit for it. This double helix, though, is these two winding, like spiral staircases. There is one strand that kind of reads from 5' to 3', and another strand that reads in the opposite direction. So we call one leading and call one lagging when we're talking about replication. Our base pairs, like I said, they go together. So adenine always goes with thymine. In RNA, adenine goes with uracil. And they're held together with 2 hydrogen bonds. You can see your hydrogen bonds here and here. And guanine and cytosine are actually held together with 3 hydrogen bonds. And so depending on your pair, you have a different number of hydrogen bonds that will occur between those 2 pairs. The connection here is what makes the helix structure. So again, this is just kind of reiterating all of what we've been talking about. Again, another reiteration with some more pictures kind of showing you, like, here's the hydrogen bonds that hold the DNA together. So go ahead and try it. So write the complementary base sequence. So if you are matching one DNA to another DNA, what would the sequence be? Alright, did you get it? So all your A's should be bonded with T's, your G should be bonded with C's. There are several simulations that I've posted in Canvas, some really great like little practices to match them together. Quick practices are really good. If you feel like you're feeling a little like I don't really get it, go to one of those simulations to help you like play. It's like a little game you're playing to put it together, and that can be really helpful just to cement those ideas in your head. So DNA replication. What happens is you separate the DNA, okay, an enzyme called helicase, okay, helix case for enzyme, helicase, breaks the it breaks the hydrogen bonds up, so it unwinds the DNA, okay. Then another group of sugar phosphate backbones and nucleotides actually come in, and they pair up with the two strands that have been broken apart. And then so you break apart one strand of DNA, or one helix of DNA, and then into two strands, and you copy everything, and then you zip the DNA back up, and now you have an extra copy of it, and that gets zipped back up. So your DNA is totally replicated, and this allows us to undergo cellular division. So kind of here's an image of that. What they don't show you here is the helicase doing its thing. The helicase would be here kind of unzipping it. And then you've got your new DNA strand builds on your old DNA strand. So your new DNA strand is now kind of like a template for building this whole double helix again, and so it produces two exact copies of this DNA that then can strand together and make its own thing. DNA polymerase, okay, makes a polymer, and it catalyzes the formation of those bonds between the nucleotides. And so you've got kind of this copy made of the base, and then you've got the nucleotides are put together. So another enzyme comes together and makes sure that all the links are there. They make sure everything is linked together and not just like single nucleotides. It makes sure all the nucleotides get connected. You get daughter DNA from that. So your parent DNA strand comes out, and you get two daughter DNA strands that you can then put together to make a new parent for the next time it replicates. So do a quick learning check. How many hydrogen bonds link the following pairs? Do you remember? Right, A and T have 2, and C and G have 3. Alright, so my cell can replicate its DNA, so it can undergo cellular division. But that's not all that my DNA gives me information for. My DNA gives me information coded to make proteins. And so we're going to go through the whole process of how I go from genetic information to proteins, because the purpose of the DNA is not to make, it's not to split and to make new cells. I need to split to make new cells, right? I need to undergo cellular division. But the whole point of having the code in the first place is to code for all the things I need to keep my myself alive, right? So I need all of the code to produce proteins, and those proteins go and do all kinds of stuff in the body that you know. So here's a picture of something called tRNA, okay? And we'll talk about tRNA and a bunch of different this is tRNA and mRNA. And we'll talk about these guys. So what is RNA? RNA is actually makes up the most of the nucleic acid. So we think maybe DNA is like the primary one, and then no, actually RNA is more than DNA found in the cell as far as nucleic acids go. RNA does all the work. So DNA gets this whole like, oh, DNA is the most important thing. DNA holds the code. Okay, but RNA is doing all the work to actually make the stuff. DNA doesn't make proteins. It's the RNA that makes the proteins. And so RNA is different than DNA. We talked about the ribose versus the deoxyribose. Uracil replaces thymine. RNA molecules are also single-stranded. They're not double-stranded. They don't need to be protected by this double strands, right, in the nucleus of the cell. They're actually going and doing work, and so it's easier for them to be single-stranded. And they're a lot smaller, so it's not like a bunch coiled together. Like, you're storing information in DNA, and the RNA is the using of that information, and so it's going to be a more efficient molecule. So you've got 3 different types of RNA. You've got mRNA, which is messenger RNA, and that does exactly what you think it does. It carries the genetic information from the DNA in the nucleus to the ribosomes. So messenger RNA goes into the nucleus, and it copies the DNA, and then it goes out into the cytoplasm out of the nucleus, and then it starts working. Transfer RNA interprets the mRNA and brings the amino acids to the ribosome. And so the transfer RNA is actually connected to an amino acid, and you'll see in a second how it puts together the amino acids for that primary structure of a protein. And then you have ribosomal RNA, rRNA, and it is actually the most abundant type of RNA, and it is combined with proteins itself to make ribosomes. And so RNA is doing the work not only by sending the messages, by pulling together the amino acids where they need to be, but also is part of the protein where the reaction of this all takes place. And so RNA is really important in producing everything that we need. So here you can see the percentage of RNA and a quick little chart telling you what the function of that RNA is. So tRNA, this is your transfer RNA. So your tRNA, they have an acceptor stem with a nucleotide sequence ACC, and then it contains an anticodon. The anticodon is 3 specific bases that complement on an mRNA. And so our anticodon is at the bottom here, and then you have this kind of, think about it as like a T shape to help you with that transfer RNA. I mean, it's probably more like on the right where it's kind of like messed around, but you can fold it out into a nice little T shape. And then you have the acceptor stem, which is always an ACC. So protein synthesis is going to start with transcription. So we're going to start with DNA in the nucleus. We're going to make some RNA in the nucleus from that DNA. That's called transcription. Okay. So transcribing directly from the DNA to make the RNA, which is mRNA. That mRNA then goes out into the cytoplasm, hooks up with the ribosome. That ribosome reads the mRNA and connects the mRNA with a tRNA and converts that to the amino acid to make a protein. So that is your translation. Okay, so transcription is where we're kind of copying down the information, transcribing it, and then translation is we're taking that information and actually translating it into a protein sequence. And so you can see here in the nucleus, you're making your RNA, and then your RNA travels out of the nucleus and it gets translated into protein. And so transcription, we unwind this DNA, right? And we do that with DNA helicase. And then RNA polymerase enzyme, which means it makes a polymer for RNA, it uses one of those strands as a template to make mRNA. And so it's just the same as we're talking about DNA replication, making that template, except now every time I see an A, I'm going to put a U instead of a T, because now I'm talking about RNA, which has no thymine, only has uracil. And so here's another image of our RNA polymerase going through, the enzyme going through and making that mRNA strand. So can you pick ABC multiple choice? Which strand of RNA goes with this template of DNA? All right, your answer was B. Make sure that you're not using any T's in RNA, only U's. The rest are the same. So now we're going to move to translation. Translation is, I've got the mRNA, okay, and I've got it into this protein thing called a ribosome, okay, and it is gonna be translated into an actual protein primary structure. The anticodon is very special, and it only bonds to 3 codes, 3 letters, we call that a codon, on the mRNA. And so the mRNA, if the mRNA is UUU, then the tRNA, right, would have a sequence that is opposite of that. So it would be TTT, and that would code for PHE in your amino acid sequence. So you have a codon for each of your 20 amino acids, and again, I will not have you memorize that. I will make sure that you know that. Your codon, AUG, starts the protein synthesis. So what the ribosome is going to do, it's going to go until it finds AUG, and it's going to say, okay, we're going to start here. And then there are 3 different stop signals, UGA, UAA, and UAG. And it'll go all the way until it reads one of those stop signals, and then it will stop. So that protein line will be done when it reaches those. And so you're just going to read the base of the mRNA as your code, not what would be on the tRNA, because the like you see this picture up here. The tRNA is going to be opposite, so we call that the anticodon. You're only going to be reading the mRNA codon to code for your amino acid, even though technically the amino acid is on top of the tRNA up here. It gets connected up here. You're only going to look at the mRNA codon, not the tRNA anticodon for that specific code. And so here are all of your codes. I will give you this on your exam. So you've got your start and your stop codes, and then you've got each of your amino acids have a code or multiple codes depending on which amino acid it is. And so if I give you a sequence like this and I give you this chart, you should be able to say, okay, here's the amino acid sequence that would follow. And so CCU is proline, and so where we see CCU, we would say PRO. AGC is serine, so where we see that, we would say SER. GLY, glycine, GGA is glycine, so where you see that, we'd put glycine, and CUU is leucine, and we would do that here. So how we read that is, you're looking for the code, your first letter on this side, your third letter, your last letter is here, and so you can kind of narrow it down that way. So if I give you these codes, what is the order of the amino acids that would go here? So give it a pause and try it. All right, so we should see that first we have GCC, which is alanine. Then we have GUA, which is valine, and we have GAC, which is aspartic acid. And so the tRNA has an anticodon, which is the opposite of the mRNA codon that you just read. Okay, and then you have an acceptor stem on the other side that makes an ester bond with that amino acid. And so you get that tRNA synthesized when you make that ester bond between the acceptor stem and the amino acid. And so we'll go back up and check out that acceptor stem really quick. There we go. So here's your acceptor stem. And so we're linking together this acceptor stem, the serine here, with your actual amino acid part. So, oh, they have it down here too, which is cool. Oops. And then you've got your mRNA binding to the ribosome. You get your tRNAs to come down, and then it's got like a little bay for your next tRNA to move over. And so the next tRNA comes down. So you can fit 3 tRNAs kind of in a ribosome. Imagine you could fit the those at a time. And so when the 2 amino acids on either side of the tRNA, so you've got, I forgot to draw. You've got like, so here's your, that's not right. So you have your tRNA, it's got an amino acid up here. The next one comes in, it's got an amino acid here. And so these are going to be close enough together, and they're going to move in a direction to connect these guys. And so then you're getting these long chains popped out on this side. And so your tRNAs are putting your amino acids together, and amino acids are held together with a peptide bond between a C-terminus and an N-terminus. And so you know that from our previous chapter. And so you start with a tRNA, it does its job, and then it shifts to the next one. We call that translocation. And so we're actually shifting places. I have a really great simulation on Canvas that shows you this happening, like this an animation that shows you. So I definitely recommend you watch all the animations under this chapter for Canvas to get an idea of what's going on in there. And so we do chain elongation, which literally means just like builds the rest of the peptide chain. And so we just kind of keep growing, kind of keep pooping out those amino acid chains. So here's kind of the process. So we start in the nucleus with our mRNA. We make that mRNA copy. That mRNA comes on out, finds a ribosome, and the ribosome has places for the tRNA to come in and bond. The tRNA has amino acid on the end, and so you get the tRNA come in, and then it gets moved over, and so these two get connected. And then once the tRNA is done, it gets popped out. It doesn't have an amino acid on it. So now this tRNA is going to go find another amino acid to put on this end, and it's going to start the whole process again. So here you can see your amino acids and your tRNA are just kind of floating around, and because of their hydrogen bonding and because of those ester bonds, they'll be able to come together, or sorry, those peptide, yeah, the ester bonds that hold those together will be able to make new bonds. And then you have a stop codon, okay? There's no tRNA for the stop codons, and so the ribosome will try to find a tRNA for that, and it won't. And so then you don't get anything happening that last step, and the polypeptide is just going to detach from the ribosome, and it's going to just go into the cytoplasm. It's going to do its thing then. It's going to make its secondary and tertiary structures to become its own protein. And so, yeah, once that polypeptide is released, then you get those structures. The protein will just build itself. After you get that long chain, because of the hydrogen bonding and the hydrophilic hydrophobic interactions, those disulfide bonds, it'll just mold itself together into a tertiary and quaternary structure, making it a protein that actually works. And so here's your quick steps into the protein synthesis, all in nice one long thing. So this is just giving you these complementary sequences. So if you're doing DNA information versus DNA template, it's like a daughter DNA strand. If you're doing ribosome, here's what that looks like. And if you're doing anticodon work, that would be the opposite of the mRNA. But I want you to use you should always use the RNA codon that codes for the amino acid. This is opposite, so it can connect these two things together. So do a quick check and tell me which part of this process each of these things are. So ribosomes moving along the RNA, adding amino acids. This is your translocation because we're changing location on the mRNA. And we're kicking out all the old tRNA that already did its job, and we're adding in new tRNA to build that peptide chain. Termination is what we call releasing that completed peptide chain after we have reached our stop codon. Activation is when you have your tRNA attached to the amino acid. This is out in the cytoplasm, not anywhere near the ribosome. And then you have initiation when you have your first tRNA binding to your start codon, AUG, on that ribosome. So let's do another check. The following section of DNA is used to build mRNA. What is the corresponding mRNA sequence for this? What are the anticodons on the tRNA, and what is the amino acid order in the peptide? So tell me all that information. Scroll back up or get out your slides and look at that chart of your codes. Okay, so here's what you should get. The mRNA sequence would be the opposite, except for instead of T's, you gotta put U's. And then the anticodons on the tRNA are opposite of what you should have put for A. But you should use what's on the mRNA to code for your peptides. So there are your peptides here. And just think about all the work that you had to put into to do that, and this is happening thousands and thousands of times a minute in our bodies, all in every single one of our cells. Antibiotics can actually inhibit protein synthesis sometimes because they stop bacterial infections from interfering with the synthesis of proteins. And so you want to keep your bacteria from synthesizing proteins. And that will kill the bacteria. And so here's some different antibiotics and how they actually get rid of the bacteria, and they keep the bacteria from doing its thing, synthesizing its proteins. If you can't synthesize your proteins, you'll die, so it's killing the bacterial infection. Have you ever taken any of these? And that's what it's doing to the bacteria in your body. So see if you can look at these and tell me what, put them in order. So see if you can put A through E in order as far as protein synthesis goes. So pause and do that. All right, here's your order. You should have gotten E. First, you have your DNA make mRNA, and then your mRNA attaches to a ribosome. That tRNA brings an amino acid to the mRNA in the ribosome. Then the ribosome moves along, translocation, and then you have your polypeptide released. And then you have your protein being built from that polypeptide into your secondary, tertiary, quaternary structures. So mutations. How do we get mutations? We get mutations from things going wrong in this process. So there's a mutation that doesn't produce melanin, that makes albino animals, and then people as well, not producing melanin. So here's an albino peacock, really beautiful. Mutations, a change in the sequence of DNA, right? And this can happen anywhere in this line. So if you wind up with a change in that sequence of DNA, you changing the sequence of the amino acid, right? So changing one of those letters is going to change what amino acid is there. And then if your amino acid has a different R group, right, then now I have different hydrogen bonding and hydrophilic and hydrophobic interactions, and my protein looks different. And my protein looks different, got a different structure, it might have a different function. It might not work the way it's supposed to. You can have mutations in your DNA result from X-rays. Any radiation can expose you to risk of mutations. Chemicals called mutagens can mutate, change the nucleotide sequence, and then there's some viruses that can go in and they can cut up DNA, and they can insert things in DNA as well. A somatic cell is a cell that's not a reproductive cell. And so if you have a mutation occur in a somatic cell, not a reproductive cell, the DNA is limited to that cell and daughter cells. And so like if you had a mutation in one of your somatic cells, that would only affect you. But if you have a mutation in a germ cell, which is a reproductive cell, then any children that you would have would also carry the mutation. And so when we talk about disorders being genetic, that's what we talk about. We're talking about there was a mutation at some point in the gene line, in the germ line, that something that happened, then that got reproduced into all of the offspring of that person or that animal or that whatever we're looking at. And you get cancer from mutations if your mutation is causing uncontrolled growth. So you have a few different types of mutations. A point mutation is a replacement of one base with another. So we talked about this when we talked about sickle cell. Right? So if you're taking one R group with another R group, or one amino acid and another amino acid, that's a point mutation. It changes the nucleotide, which then changes that different amino acid. And so that different amino acid now makes the protein shape differently, which, like we talked about before, it would make like sickle cell. If you have a point mutation that changes it, right, we call it a point mutation. But you can also have a point mutation that doesn't change the amino acid sequence, and that's called a silent mutation. So if you look back at that amino acid chart, you'll notice that there are several codons for one amino acid. And so if you're just changing between those, it won't actually change the amino acid sequence. So if you're still serine at the end of the day, even if you change the letter, it's still going to code for serine, and then the mutation is only from, like, it doesn't affect the actual building of the protein, and so we call it a silent mutation because, like, it's not going to affect the function of that protein. A deletion mutation is when you just take a base and it just gets totally deleted in the strand. And of course that would result in a different order of nucleotides, and then an insertion is just a base being inserted, and of course that changes it as well. So normal DNA sequence is producing that mRNA, right, which then gets translated into amino acid sequence, which then goes forward and bends and twists into a protein. If you have a point mutation, we have, if we change C to a T, then we change glycine. So glycine was here. We're changing this C right here to serine. And that's the wrong amino acid in that chain. So it's going to mess up that protein. Right? That's a point mutation. A deletion mutation looks like this, where we just take something out. And so now, where I'm supposed to have this order, everything now shifts down, and I get both of these are now coded incorrectly. If I get an insertion, it's kind of the opposite. So now everything, of course, is going to shift, and now this is my 3 instead of AAA being my 3, right? And so you're getting still a difference in these amino acid sequences. Your mutations can cause the changes in the proteins, causing a loss of its biological activity or making it behave differently, and that really messes up the whole system. When we have this system it's hereditary, meaning it's in the germ cells, we call it a genetic disease. If it's only affecting the somatic cells, then that's typically a cancer. So genetic diseases are caused from a mutation in the genetic code. Albinism, phenylketonuria, PKU, results when the DNA cannot direct the synthesis of a certain enzyme. And so you need enzymes to do a whole bunch of different things, and if you can't make that enzyme, then you can't do that thing, and of course you'll get something that is affected by that. So for this example, they're telling us that albinism results when that enzyme that converts tyrosine to melanin is defective. But if you have another one, then you can have a different disorder. So here's a list of some genetic diseases and their results, and where the mutation is. So if you're interested in any of these, definitely check out this this chart. So go ahead and tell me if it is a point mutation, a deletion, or an insertion. So cytosine entering into the DNA sequence, that's an insertion. Adenosine removed, that's a deletion. And then changing is called a point. And so there's something called recombinant DNA that geneticists use in engineering. Recombinant DNA is what's formed when we cut and combine DNA fragments. We can put DNA pieces together like puzzles, and we can cut, snip, and engineer DNA, which is pretty cool. So to prepare recombinant DNA, you need they try to do this in Jurassic Park. If you've ever seen like the original Jurassic Park, they say like that they put together these like pieces of dinosaur DNA with, what was it, like frogs or something. That's recombinant DNA. Okay, so you take a fragment from one organism and combine it with another to like complete a sequence. And then you've got all of these things that you don't need to know how exactly this is all done unless you're like really interested in going into it, but I'm not going to have you do this on the exam. It's really interesting because we use DNA plasmids in E. coli to piece together and like to pick out these things. And then we join them together with an enzyme called, enzymes classifications. Ligase is an enzyme classification. And then you put that new gene and put it into the whole process and makes protein. And so here's kind of your image of what we do with recombinant DNA and what we do. So it's like cloning. So we take these plasmids, these E. coli, we isolate them, we cut a piece out, we put the other DNA into the plasmid. So bacterial cells have really simple DNA strands on these like double helixes, and so we're cutting into that. So we have like this very scientific term, sticky ends. With the sticky ends, we put the donor DNA in here. So now we have the new DNA. This is recombinant DNA. And then we start replicating. And we start making new cells and clones and new proteins using this DNA. So it's really easy to do in bacteria, but we have been able to do it with more complicated systems, right? That's kind of the idea of cloning. But I mean, like designer things, like we actually use something, there's a new system called CRISPR that's being used. Definitely check out information about that if you're interested in all of this. But it allows us to really like take out and fix problems and mutations in DNA, but also allows you to kind of put in, replace things maybe that don't need fixing. So there's a lot of ethical things that go along with thinking about messing with DNA like that. But there's a really great treatments for using recombinant DNA. So you got a problem with your DNA, well, I mean, we can fix it. If you have a problem producing insulin, we can help you do that. We can use recombinant DNA to help with destroying tumors, with helping people who have hemophilia. If you've ever gotten a flu shot, that comes from using recombinant DNA, and so we can't have the good without maybe the bad, but hopefully the good uses of these sciences will outweigh any bad that people might think to do. Like life-saving things that we can do with genetics now. So a polymerase chain reaction, or PCR. PCR is something that we use a lot in testing. And so like if you are a crime scene investigator or forensic scientist, you might find just a few strands of DNA in a sample, and you need to test it to see what it was, but you don't have enough to test. And so we can do something called PCR, and we can make a larger sample of the same DNA so that we can run it through other tests. And so if you ever work in industry, food industry, you may wind up doing this. I did PCR working in the pecan processing industry, and I would be testing for salmonella bacteria. And traces of salmonella are bad, but I need to know if there are any traces. And so I would test for the salmonella DNA by putting it in a PCR machine and multiplying whatever DNA I had, and then running my test for salmonella to make sure that I did not even have any trace DNA of salmonella in there. And so if you test a larger sample like this for this small sample, then you get no hits here. You know for sure you didn't have any of this, right? And so that is very helpful to make sure we don't have, or make sure we do have something that we need to have that maybe only happens in very small regions of DNA, not enough for me to actually test. So we can screen for genes. This is how we screen for BRCA, and how any genetic testing really is done. PCR is typically done on that, just to make it enough for you to actually see the results of that DNA sequence in a test. You can also do DNA fingerprinting. We can take a DNA sample, cut it into fragments, and we can use electrophoresis to separate the fragments out. The banding pattern, and so this guy's holding electrophoresis banding pattern here. This banding pattern is called a DNA fingerprint, and everybody has a different DNA pattern because everybody has a unique DNA sequence. The human genome was finished in 2003, and so we have a generic genome totally coded for humans, which is really great. It is all of the bases that actually code for protein, but that's only 3% of our DNA. So there's a not correct term saying like, oh, junk DNA. It only like our DNA has a job. It's not junk. Only 3% of it actually codes for proteins. We have since found structures of DNA that actually code for RNA molecules, but most of our DNA is at regulation, right? It serves as recognition sites. It there's a function to our DNA. It is not junk. There are lots of some DNA. The only thing it's supposed to be there for is just to make sure that the rest of the DNA doesn't mess up its job. So there's function to everything. There's no junk. So don't think that. There's only 3% of DNA actually codes for proteins, though. But we know all of those bases, which is pretty cool. 3 billion bases. Do a quick learning check. See if you learn anything about recombinant DNA. You do not need to know about recombinant DNA for the exam. Alright, last bit about viruses. Viruses are very simple, okay? Here's an image of a virus. We got some lipids, we got some proteins, we got some RNA, and we have what we call a core. So viruses are very small particles that require a host cell to replicate. They can't replicate on their own, so they need host cells that can then replicate. And the whole job of a virus is to like get into a host cell and just replicate. It contains either DNA or RNA, but not both, okay? And it has a protein coat. It does not have all the things to make proteins and grow. So it needs a host cell to do that, to make proteins, to grow. A virus is not alive. So if you see everything, it's like, oh, kills viruses. You cannot kill a virus. It's not alive. You can get rid of a virus, but you cannot kill a virus because it is not living. Some diseases are caused by viruses. You may have had a cold before. Here are a bunch of other diseases caused by certain viruses. So how viruses works is it attaches to the host cell and it injects its DNA. And that DNA is just picked up by our cells. Like, no big deal. So you really want to get rid of the virus before it gets into your nucleus, before it like injects its DNA, right? And so you have other proteins whose jobs in your immune system is to recognize the virus and like kill it, get rid of it, dispose of it. Sorry, we can't kill it. Dispose of it before it injects its DNA into the host cell. Or we want things that recognize that what just happened in the host cell was wrong, and then that cell gets killed off before it replicates. But what it does is it just inserts the DNA in, and that DNA just gets clipped in to our DNA, and then we replicate the bad DNA. We replicate that viral DNA, which then makes new viruses, and those new viruses go out and infect other cells. And so they're kind of taking over our system. And it will burst the cell open. It will just destroy the cell. But those new viruses are all released, so they're building up inside the cell and then they get released. Kind of scary what a virus does. And this takes you through all the process of what a virus is doing. So a retrovirus, if you've ever heard that, it contains viral RNA but no DNA. And so what happens if the virus contains RNA but no DNA? It uses reverse transcriptase to produce a viral DNA strand. So, I don't have any DNA, so it uses its viral RNA will actually produce a DNA strand. The new DNA strand will form a complementary one, because RNA only has one strand, and then that new DNA strand will go in and use nucleotides and enzymes to synthesize new virus particles. So if it doesn't have DNA, it'll make its own DNA and then do all the things to make new viruses. It's called reverse transcription because you're taking RNA to make DNA instead of the opposite way of making DNA into RNA. So here's reverse transcription. So it's your so retrovirus, like HIV, it works like this, and it makes this viral DNA, this provirus, that then gets put in, and then we make more virus. AZT and DDI, they inhibit this step. They reverse transcriptase. They don't allow that to happen. So that's why it's that those certain drugs, they inhibit this from happening. And then you also have drugs that will inhibit the virus from even entering. And you've got drugs that can inhibit the protease inhibitors to let the old viruses out. So you could trap the virus in that cell will die, but the old viruses won't be able to come out. Those are the new ones. And so the HIV virus is a retrovirus, and it infects T4 lymphocyte cells, which is harmful because the T4 lymphocyte cells are needed for your immune system to destroy other harmful things. And so it is the immunodeficiency virus because it makes your immune system weak. And so it can cause you to just get really, really sick really easily, and that's really what hurts people with HIV. And then AIDS is based on attacking the HIV at different points. So if you're trying to treat it, you're trying to get into one of those places like you see in that cell to stop that reverse transcription or to stop the virus from even getting into. At some point in the life cycle, you're trying to stop that virus from doing its thing. So here's some treatments, some molecules. You can see these different molecules that are used to treat. You can go through and match these together, match the terms. You will need to know about how a virus works. I'm going to just match the terms. A virus containing RNA is a retrovirus because it works in the reverse direction. A virus is a small particle requiring a host to replicate. A substance that prevents the synthesis of viral proteins, protease inhibitor. I'm not going to have you recognize those. And then using viral DNA to synthesize using viral RNA to make DNA is reverse transcription, which is with a retrovirus, right? We'll do that opposite, reverse transcription. So you do need to know those. I'm not going to have you know protease inhibitor. And so if you have cells that continue to grow without then you can have cancer being caused. There are tumors that are non-cancerous and that are cancerous. You can get rid of those by radiation and other treatments. There are carcinogens in the chemical environment, so like asbestos is not very great, so you should probably stay away from that. And then here's your concept map for Chapter 17.